I'm Liz and this is Liz Loves Science, and today I'm going to talk to you about electron microscopy. So I've already done a video on light microscopy, or optical microscopy as it's more correctly known. So I'm going to do a little bit of an overview on that today in case you've not seen my video, and a bit of a recap, and then I'm going to tell you about electron microscopy and why we'd need to do that. So this is my light microscope. So we've got a little light source here and the stage, and we've got our lens, our objective lens here, and the eyepiece lens or the condenser lens here, and the light goes through the sample, going through the bits that aren't so dense and getting blocked by the bits that are dense, gets collected by the objective lens and then focused onto the condenser lens so that you can see it with your eyes. So I'll draw a little schematic of that, but that's the basic fundamentals of light microscopy. So here we've got our sample stage and our light source here. Black's probably not the best colour for a light bulb, but never mind. And our sample's mounted on there. Your sample can be, well, you can look at anything under a microscope, but if you want to look at plant cells, a good one to look at is onion skin or onion membranes, because they're really easy to get out of the onion and it's pretty much ready to go. You can stain it with a bit of iodine, which you may be able to get from a chemist or online, but that's pretty much all you have to do. So the light comes out of a light bulb here and goes through our sample and is then collected by the objective lens. And this, let's have them all crossing at the same place, shall we? And this takes the light that's come through and magnifies it. And then here we've got another lens, and that's just below the eyepiece because it needs some space to focus it. Same as your glasses, focus your light on the back of your eyes, so you've got that focus space in front of your lens. And this is our eyepiece, and there's our eye looking at it. Let's give it some eyelashes. There we go, and that is our light microscope, and that's all in a dark tube so that you've not got light coming from outside to interfere. That's good as far as it goes, and as I say, you can see quite a lot of things under a light microscope, but there are limitations to it. So one of the main limitations is, due to the wavelength of light, you can't see anything smaller than about 200 nanometers. And that's an absolute value. As I say, it's based on the physics of how it works. You need to be able to get the light in between the bits of the sample in order for you to be able to see them. So that is the limit of light microscopy, which is a bit of a downfall. So, as I said, you can look at onion under a light microscope, and if you do, you'll see something like this, and you might see you'll see some dots in it. I'm going to do these as green dots because they are green. It's what gives plant cells their green coloration. And those are called chloroplasts. I'm going to do another video on different kinds of cells and what's in them. There's much more. You may see a nucleus. You're not going to be able to see much more than that on the onion cells because of the small scale that you're looking at. So you can already see some detail. You can see enough to see that there's cells there. You can see that they're plant cells, and if you're looking at, say, a sample of lung tissue, you can see the different kinds of cells in that and the different shape of them, but you won't be able to see any of the detail within the cell because it's just too small. So, in order to be able to see that detail, we need to go on to an electron microscope. And electron microscopes, there's two main types. There's transmission electron microscopes, which is TEM. Let's say that's transmission. And there's a scanning electron microscope, which is abbreviated to SEM. And these look at things that are smaller than 200 nanometers, and they look at them in different ways. So I'm going to go through a bit about how they work and what you see and how you prepare your sample to go on one of these microscopes. So, in the optical microscope, we used light going through the sample and we focused it with lenses. You can't focus electrons with lenses, so we need other ways of focusing electrons. Electrons are charged and they interact with magnetic fields, so we can use that relationship as a focusing method for focusing an electron microscope. So in essence, the transmission electron microscope is very similar to a light microscope, only instead of light we've got electrons. So here you've got an electron gun, and that fires negatively charged electrons through a sample, which is mounted on a sample chamber on a slide, and these electrons go through, and in the same way as with the optical microscope, where there's gaps in the sample, the electrons will go through, and where there isn't, they'll be blocked. So we have our electrons going through. And then here we have an electromagnetic coil. Or an electromagnet. And that works to focus the beams of electrons that are coming through. And we have another one up here. And as I said, they work like our lenses. And then this image can be focused back together on a detection plate, and that gives you your image of your sample. So a few issues with this. One is if you look down an electron microscope you won't be able to see anything. You need this detection plate, which is kind of a bit like photographic film perhaps. Currently it's done by computer, but it's a similar process to taking a photograph, whether it be through traditional film photography or whether it's through a digital camera. You've still got your method of recording where the light falls on the film. And that's effectively what it is here. In our optical microscope, everything was in a tube so that we didn't get interference from light coming in from outside of the system. And you also need to do that with an electron microscope. So this is in a chamber like this, and with electrons, they're all around us. Electrons are the outermost levels of atoms, and so in order to get this area without any electrons in it, you need it under a very high vacuum. And because of this, you can't view living cells. So under the optical microscope, you can have a look at a bacteria that's swimming around. They are used for doing samples of sperm, for example, when they're doing IVF and looking at injecting the sperm into the egg. You need to be able to do that on a living cell. With these, because living cells require oxygen, you can't do electron microscopy with it. So it's not a kind of universal technique that can be used for everything, but it does allow you to see things much smaller than the 200 nanometers. And in biological terms, you can see the components of cells in there. So it's kind of as small as we could need to be able to see. So that's our transmission electron microscope, and you may have seen kind of pictures of cells that look something like this. And those are, well, that's a drawing obviously, but those pictures would be taken with a transmission electron microscope, because these bits inside the cell are way too small to be seen with an optical microscope. So that's our transmission microscope. The other type of microscope that you might want to know about is a scanning electron microscope. And again, it's kind of similar how it works, but it's looking at things from a slightly different angle. So here you have your sample on a plate, like that, and instead of the electron gun being underneath the sample and shining electrons through it, we've got our electron gun here at an angle, beaming electrons down at the sample. So those electrons will bounce off the sample and be detected in a detector. And this is your electron gun. And how it works is this electron gun scans across the sample. So it'll start beaming beams down here, which will come back there. And by looking at the angle of the reflected light, electrons, and the angle of the electron gun to start with, and also by taking into account whereabouts within the electron's wave, because an electron has a waveform similar to light, so whether it's at a peak or a trough or somewhere in the middle, then the computer attached to the detector can tell kind of the depth and angles and things of the electrons bouncing back. So that's all needed to be put together, which is done computationally, and the image calculated. So this one is a bit more, a bit less obvious how it works, but basically you're shining electrons on it and you're looking at what bounces off, a bit like if you look in a mirror. So I'm here, with my glasses on. Oh, my nose is going the wrong way, never mind. That's me. It's not a very good picture of me, but never mind. And here's the mirror. And in the mirror, so we've got light bouncing off my face and going into the mirror, and then it's bouncing off the mirror and going back into my face so that I can see what I see. And my brain then processes the image to turn it into something that I recognise as being an image. So this, again, it needs to all be done in a vacuum to avoid the problem with the electrons in the atoms of the atmosphere that you get with the transmission electron microscope. So again, you can't do anything that's alive, and you tend to need to stain this, and how you would do that is by spraying it with a very fine coating of a heavy metal. So sometimes gold's used, and that enables the electrons to reflect off the surface better, and it prevents it damaging the sample. So if we draw this in a more upright thing, so we've got our chamber here and our sample down here, and we've got our electron gun blasting electrons down, and they're coming up, and they are then seen. They're done using a detector like this, but as it scans across, you get a dot there, and then if it goes there and there, you get a dot there, and there, until it's built up a picture, and the electron beam goes across, making little dots of electrons on the detector. So you'll get something looking like and there you can see there's starting to be a picture built up, something kind of this shape, maybe a pair of tights or something, and that's how the scanning electron microscope works. Again, you may have seen images taken with the scanning electron microscope. If you've ever seen, the one that you tend to see quite frequently is an image of a fly's eye, and if you've ever seen, or a fly, if you've ever seen an image of a fly looking really huge with big hairs sticking off it, then that's been taken with a scanning electron microscope. As I say, you can't look at things that are alive because there's no air in the chamber. This is again a high vacuum. But you can get a good image of the texture of something, whereas the transmission electron microscope looks through your sample. So that's how electron microscopy works. As I say, advantages are you can see much more detail and you can see much smaller things than you can with an optical microscope. The disadvantages are that you can't see anything that's alive and you need more sample prep. And also, as I say, I have in my house a little optical microscope. It's small, but it does the job and it's pocket money. You can get better ones still for not vast amounts of money. Whereas if you're wanting an electron microscope, you need an entire room for it, and the sample preparation takes some time and is a skilled task, and these are millions of pounds worth of equipment. So there are advantages to each system. It depends what you're looking for and what you're wanting to see. So that is my brief overview of electron microscopy, and we're going to go on then to look at cells and what these things that you can see within the cells are. So that's going to come up in a future video. I hope you've enjoyed this one. If you've got any comments or if there's something you'd like me to see, then leave a comment in the comment section. Or if you like what I'm doing, then hit the like button and subscribe to my channel. See you next time. Bye!